All right. I wanted to start class today by talking about the final exam, which isn't too far away. So we're taking it December 18th, like I've mentioned before. It is semi-cumulative, which means a percentage, about 30% of that grade will come from your previous knowledge in the past 3 units, but most of the exam will focus on new material, right? I know that can be daunting, right? So what I have done is I have linked a sheet up on Moodle, these semi-cumulative questions on the final exam, right? And what I'm going to do for the semi-cumulative portion is I'm essentially going to give you 4 of these questions, right? So you get the questions ahead of time. There's 10 questions on there. I will show you 4 of them on the exam. Right? So this is a way to focus your studying and kind of the big concepts and not get bogged down by every little detail we've talked about in the past 3 months. Right? So this should be super helpful. Right? But because I'm giving you the questions ahead of time, I do expect really complete and detailed answers. Right? So I'll be grading them a little bit more harshly just because you've had time to use your notes, textbook, lecture slides, whatever you have to answer these. Right? I'm not going to grade these ahead of time, so if you give me your questions and say, can you look them over and make sure I answer them correct, I'm not going to do that, but I can, answer any clarifying details, clarify any concepts of, if you have any questions about these, let me know. I will pick 4 out of this list, and each question on the exam will be worth 5 points. All right. Any questions in general about this? All right, so when you're studying cumulative material, focus on these questions. Don't get bogged down by every little detail we've ever talked about. All right, let me move over. All right, we're gonna do a little bit of math today because we're gonna talk about population genetics. And so population genetics are really looking at the allele and genotype frequencies in a population. So I know we've talked about what is a population, right? And we have so far learned this definition, right? It's a subset of individuals that occupies a particular geographic area and can reproduce, right? So I just want to emphasize a couple of these points, right? That it will be a group of sexually reproducing organisms, right? So organisms that asexually reproduce would be genetically identical to each other. We don't really consider that a population per se, but rather a group of identical organisms. Right? They're going to be able to interbreed. Right? They can reproduce, which means that they're able to interbreed. It's going to be a single species we're talking about. Right? So population will be a single species. If we're going to talk about every species in that geographic area, then we would be talking about the community. Right? So population, single species. And finally, they occupy a geographic area, right? Which means they're going to occupy an area that's easy traveling distance for them, right? And that really depends on the species we're talking about, right? Now, for plants in the Joshua tree here, it's really going to depend on how they pollinate and how far the pollinators travel, right? But if we think about organisms that can move, right, the geographic range of mice is going to be a lot smaller than, say, a human population. Right? We have the ability to move further distances. Just like these coyote populations here, right, they have large geographic ranges because they can move quite a significant distance in their geographic range. All right, so population. We're looking at a population and we're talking population genetics. We have to think about the gene pool. Right? So the gene pool is all of the alleles for every gene in a given population. Right? Now, typically, if we were to talk about a gene pool, we're not going to talk about every single gene that ever exists. We're going to focus on 1, maybe 2 genes at a time, which is what this image up here is doing. It's focusing on this gene A, right, which determines the beetle color. Right? We are just looking at the gene for color. Even though technically the gene pool would encompass every single gene that these beetles have, We're just looking at a subset of those genes, in particular the one gene for color, right? And so when we're looking at gene pools, we can start to look at the genetics of a population, right? We can study genetic variation within a population and how that variation might change from generation to generation. Right? We kind of see some of these mechanisms of change, the selection and genetic drift. Right? But if we're looking at population genetics, we're really focusing on the alleles and the genotypes and their frequencies in the population. All right, before we get into the math part, I just want to emphasize, right, that genes are usually going to be polymorphic. Right? And this is not a new concept, but maybe a new terminology for it. Right? Polymorphic just means there's two or more variants for a gene. Right? So think two or more alleles. Right? Everything we've looked at basically up until this point, we have been looking at polymorphic organisms, specifically diploid species. Right? So they have a capital letter, they have a lowercase letter. Right? Then we have those two variants. There could be more than two variants, right? But in Biology 101, we're just going to focus on the two. Once we add in a third, our equations start to get more complex. Right? So a polymorphic gene has two or more alleles. We could see a gene that's monomorphic, which means it really only has one allele. This tends to be more of the case when a population loses some of its diversity. Right? An example of a monomorphic gene might be in these birds, these corellas here. They are monomorphic in the plumage gene, right? They're not completely monomorphic, right? They have other genes that are probably polymorphic, right? But for the plumage gene, we don't see any variation. They are monomorphic, right? And just as a reminder, right, to get these new polymorphisms, to get these variations, we often have mutations that might cause these variations down the road. All right. Your textbooks calls them single nucleotide polymorphisms. Right. That's a point mutation. Right? A point mutation, we are changing out a single nucleotide that can therefore downstream change that variation. Yes. Oh, thank you. I'm getting really bad at this at the end of the semester here. I appreciate the reminders. All right. Any questions on polymorphic genes? All right, we're going to move into the math portion. So we need a way to analyze the genetic variation in a population. And we're going to do this by looking at frequencies. Right? We're going to look at allele frequencies. So in my image up here, I have deer. They have two alleles with fur color, right? Big A or little a. Right? Little a is the recessive gene, and if they're homozygous recessive, we get these albino fur colors, right? These white fur colors. But we can also look at genotype frequencies. All right, so now we're looking at, are they homozygous? Are they heterozygous? Are they homozygous dominant or homozygous recessive? And what's the frequency of each of these in a population? All right, now for the math. So we want to calculate an allele frequency, right? The basic formula, and again, we're just focusing on that single allele, right? So the allele, not the genotype. We would look at the number of copies for that specific allele in the population, right? So maybe we're just looking at the allele frequency of the capital A, right? And we're going to divide that by the total number of all of the alleles in the population. All right, that would be the basic formula. We'll put this into practice on the next couple of slides here. All right, the genotypic frequency. All right. Same kind of concept. Instead of looking at specific alleles, now we're looking at genotypes. So it'd be the number of individuals with that particular genotype that you want to look at. Right? So maybe it's heterozygous individuals, divided by the total number of individuals in the population. All right, so these are your basic formulas here that we will apply. And we're going to do this with an example. All right, so we have this population of these plants. They're called 4 o'clock plants. Right? We have 100 individuals in this population, and we know how many individuals are in each genotype, right? It's a bit of a side note here before we continue. The genotypes we see here are a little different, right? We don't have a capital and a little letter, right? Why are these alleles represented as both capital letters? Does anybody have a hypothesis as to why this could be the case? Yeah? Yeah, they have co-dominance, right? So if we look at the phenotypes, we have red, we have pink, we have white, right? Those heterozygous individuals, the red and the white alleles are blending together, which is why we see two capital letters instead of a capital and a lowercase, all right? It could have very well been an example with a capital and a lowercase. The same concepts are going to apply, all right? So we have this population of 100 plants, all right? We know that 49 of them are red, 42 of them are pink, and 9 of them are white, right? So we know the genotypes for the red is RR, for the pink is RW, and for the white is WW, right? I want you to keep this in mind as we work through these examples, right? Remember that the homozygous individuals have 2 copies of a given allele, right? So either both capitals or both the same letter in this case, right? And heterozygous have 1 copy of each allele. All right, so this is where we're going to put it into our equation. So we're going to look at the frequency of the white allele, or the W allele. Right? And to do this, we want to look at the total number of that specific allele in our population. All right, now if we're thinking about the W allele, we have 2 genotypes that have the W allele. Right? We have the homozygous genotype of WW and the heterozygous type of RW. Right? So there's 2 genotypes that have this allele. Right? Bear with me here. So if we look at how we would lay out our equation, we would say RW would need to be up top because we need to account for that W in the genotype. We also have WW, and we're going to multiply it by 2 because there's 2 of those alleles in that genotype. All right? We're not multiplying this by anything because there's only 1. Then we're going to divide it by the total number of all the alleles in the population, which is essentially multiplying each genotype by 2 because there's 2 alleles per genotype. Right? So if we were to plug in the numbers here, right, for the R and the W, we have 42 individuals. So we go 42 plus, right, 2 times the number of homozygous individuals for the white allele, which we know is 9. Right? The bottom equation, we would just plug in 2 times all the numbers for the genotypes. And if we did the math and we simplified it down, we would see that this is 60 divided by 200, which is 0.3, or 30%. Right? So the frequency of white alleles in this population is 30%. Right? Just as a side note here, I know it's daunting. I have put up some practice problems up on Moodle so that you can work through several examples and practice this skill. All right, now that's the allele frequency, right? But we also have the genotype frequency. I actually think the genotype frequency is a little easier, right? Because you don't have to account for 2 alleles and looking at one specific allele over the other, right? We look at the frequency of the genotype WW, right? I would look at the total number of that genotype, which would be 9, right? Divided by all the plants in the population. So I would add all my numbers together. You know, there's 100 individuals, so it should add up to 100. I would divide 9 divided by 100, and I'd get 9%, or 0.09. All right, so that was a little bit easier. We only have to look at one genotype versus for alleles, we have to look at all the genotypes that have that allele. All right, let's step back. I'm going to have you put this into practice. We looked at the frequency of the white allele, and I want you to look at and determine what is the allelic frequency of the red allele. There are a couple of ways that you can do this, right? So think about it. I'm going to give you a couple minutes here, and I'm happy to answer any questions you might have. That's just all the number of alleles. I see some of you calculating away, so I'll give it another minute here. I get the sense that the majority of you have come to a conclusion. So what have we calculated out the frequency of the red allele to be? I'm hearing different numbers. I hear 51, or are we some other numbers? 70? I'm going to say, yes, it is 70%. Yeah. So you could have done this out just like we did this formula for the white allele, right? The only difference here is that where this is homozygous for the white allele, you would just substitute this in for the homozygous genotype for the red allele. So that would be a 49 instead of a 9. Right? You could have done it all out longhand and come to 70%. Or, right, if you think about it, if 30% of the population is one allele, right, and it's only a 2 allele population, the other percent has to be 70% to account for 100% of the alleles in the population. Right, so that would be like the simple way of doing it, which I purposely did not tell you that ahead of time. So I wanted you to maybe work through that more complicated problem. Any questions or concerns? All right, we're going to continue on then. So we're going to take what we know about population genetics and apply it to the Hardy-Weinberg equilibrium. Right? So this was proposed by two individuals, Hardy and Weinberg, just the name, right? And it describes the relationship between alleles in a population that is not evolving, right? Not evolving being the key here, right? And so if a population is not evolving, we would predict that the alleles and the genotype frequencies would remain the same generation after generation. right? So a population is not evolving, which means, right, no new mutations are occurring, right, which would increase our genetic diversity. There is no natural selection happening, right? The population is large, which means we're not going to see genetic drift or like fixation or loss in allele. Right? No migration is going to occur, so no gene flow, and there's going to be random movement. Yes, Kendall. It's a big question, right? In reality, populations rarely reach this equilibrium. All right, this is really hard to do, all right, because this just isn't the way that environment acts upon a population, right? So if this is hard to accomplish, right, why is this a useful equation? Right? Well, it's useful in the sense that if we do this equation and we realize that our frequencies are not in equilibrium, we know that there's some evolutionary mechanism in play. We don't know which one, that would involve further research, but we know something is happening in this population. This population is not stable from generation to generation. There's some genetic variation happening, whether it's due to natural selection, genetic drift, gene flow, what have you. Yes? You know, I guess it was a mathematical model rather than finding a population. Um, Honestly, I don't know of a population offhand that is Ann-Hardy-Weinberg unless it's manipulated by humans. So, for example, a lot of like lab mice might have the same genotype and frequencies, population after population, because researchers want that stability. I don't know of a natural occurrence of this. Right? But this is a useful equilibrium nonetheless when we try to look at the frequencies in a population. Right? So again, we're not evolving here. So they came up with two equations here, right? Looking at the allele frequencies and the genotype frequencies, right? So P plus Q equals 1, or P squared plus 2PQ plus Q squared equals 1, right? Now, in these equations, right, P is going to be one allele, and Q is going to be the other allele, right? So in this equation, this only works when we have two alleles for a gene in that population, right? So for example, if we had two alleles, which would be capital A and little a, we could assign P to big A and Q to little a, right? If you look at problems online, you would often see that the P is often assigned to the dominant trait or the capital letter, Q is assigned to the little letter or the recessive trait, but it really doesn't matter how you assign them. All right, again, we're gonna work through a problem together here. We're gonna work through a couple of problems together. So we're gonna look at our flower example again, all right, our 4 o'clock flowers. We already know what our allelic frequencies are, right? We already calculated this out. We know it's 0.7 for R and 0.3 for W, right? We're going to try to figure out our genotypic frequencies by plugging these numbers into our equation, right? So we're going to say P is the R and Q equals the W allele, right? So those are how I'm assigning it. If you were to assign it flip-flopped, you would get the same numbers in the end. All right? So we're going to say P squared is the genotypic frequency of RR, right? So RR, we have two alleles, right? So the P squared is RR. The 2PQ, right? P and Q, two different alleles, means we have a heterozygote, right? So that would be the frequency of the R and the W. And then Q squared, you would see, what you would say is the frequency of the W homozygous gene. Right? So that's how we're going to assign our genotypes in this equation. Right? Now we know if P is 0.7 and Q is 0.3, all we really have to do are plug these numbers into our equation to figure out these frequencies. All right, so the frequency of RR, or P squared, right? We would go 0.7 squared, right? We're just plugging that number in. We would get 0.49. Right? Again, for the frequency of the heterozygous, we're just plugging in the numbers. So 2p cubed, you would take 0.7 times 0.3, multiply it by 2. You would get 0.42. Okay, so that's for our heterozygous. And then for WW, right, q squared, you would take 0.3 squared to get 0.09. Right, so in this case, it's a matter of simply plugging in the numbers and doing the math. Right? Now, if we were to take a closer look at this, right, we calculated out the frequencies of our genotypes, but what does this mean in a population, especially a population that's reproducing, right? Well, basically, we're calculating out what the zygote probabilities could be, right? So if we were to mate two heterozygotes, and we would want to mate two heterozygotes in this case, because if we didn't, we would not get all the possible allelic combinations, right? But if we were to mate them together, we could say the probability of making an RR zygote would be 0.49, which is exactly what we calculated, right? The probability of making a zygote that is RW is 0.21, right? Or WR is 0.21. Now notice 0.21, we did not calculate a 0.21, we calculated out 0.42, right? But there's two chances of making a heterozygote here, depending on which parent is giving which allele, right? So we would simply add these together, and that's where we get that 0.42, right? And then that probability of making a WW zygote, right, is also what we calculated out as 0.09. Right, so this is kind of the mechanisms in play when we're talking about this equation. Right? Big picture thing here to remember, right? When you are doing these and you want to check your work, make sure your work adds up to 1. Right? If it doesn't add up to 1, you know that there was a mistake somewhere in your calculations. Right? Genotypic frequencies are always going to equal 1. Same with allelic frequencies. They're always going to equal 1. Right? Because we have to account for 100% of the population. All right. So this means, again, in Hardy-Weinberg, no evolution is occurring. Right? When we look at the gametes, the zygotes, the young individuals, the adult individuals, the probability of grabbing an individual with a certain genotype and the probability of grabbing a certain allele out of the population is going to be the same, generation after generation. Right? Nothing is changing. All right, so how do we look at genetic variation? Well, we look at the allele and the genotype frequencies, right? And we can do this by using our Hardy-Weinberg equilibrium equations. Are we ready for another example problem? All right, so this one is a bit more complicated because we don't know how many individuals have each genotype right off the bat. Right? In this problem, we have a population of 200 deer. 65 of them are white. Right? From just this information, we can calculate what the allele frequencies are and what the genotype frequencies are, just by knowing that 65 of them are white. Right? So let's start by what we know. Right? So we know 65 of them are white. And white is the recessive allele in this population. So what is the genotype of the white deer going to be? Little a, yes. Right? What would the genotype of the brown deer be? Yeah, we have those two possibilities. It could be big A, or big A, little a. Right? We don't quite know what their genotypes are just by looking at them. Right? But we do know the genotype of that white deer is little a, and we know that for sure because in order to be white, you have to have 2 of those recessive alleles. Right? We also know our Hardy-Weinberg equilibrium equations. Right? P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. Right? This first one here would be our allele frequency. The second one being the genotype frequency. right? We also know we have two alleles, right? Big A and little a. And I am going to assign P to be the big A and Q to be the little a, right? So if 65 of 200 of these deer are white, right, that means that 0.33, or 33% of our individuals have the genotype of being homozygous recessive, right? So that's the easy one we can calculate and get out of the way, right? We know this genotype is little a. We know that 65% of these, or 65 deer have this genotype, which in this population would be 33%, right? So genotypic frequency of little a. All right. Well, we need to know more than just that genotypic frequency. All right. So we're going to, again, use that information we have figured out so far to figure out the rest. So if we want to look at the other genotypic frequencies, and I like to rewrite my equations with the alleles, right? It helps me remember what I assigned as P and what I assigned as Q, right? I'm going to use the genotypic frequency equation because I'm looking for the genotype. Right? I can plug what I have just learned. Right? We calculated the frequency of the recessive, right? Homozygous recessive genotype to be 0.32. I can plug that into my equation. Right? I know that in order to find the allele frequency, I can take the square root of 0.33. Right? So normally it's squared, we're going to take the square root of it. Right? This is probably the biggest piece that might be the least intuitive, right? Especially if you're not a math person. Right? But if I were to take the square root of 0.33, I would find that A, just a single allele, not the genotype, is 0.57. All right, so I have now found the genotypic frequency of AA, right, and the allele frequency of A, right, being 0.57. All right, again, we're going to use what we just calculated to continue on down the line here, right? This time we can use the allele frequency to figure out the frequency of big A. We know what little a is. We know it's 0.57. We just calculated that out. So we can put that into our equation. We could say P plus 0.57, right? So I assign my Q as the little a, equals 1. Now if I rearrange this, it would be P equals 1 minus 0.57. Right? So some simple math here in order to get the p-value of 0.43. Right? So I know that P equals A, big A. So big A, the frequency of that singular allele is 0.43. Right? Now if I were to add this together, the big A and the little a, it would equal 1. Right? Because it's counting for 100% of the alleles in that population. Right? So big A will be 0.43, little a would be 0.57. All right, the last step here, because we still don't know the genotypic frequencies of the heterozygote or the homozygous dominant, right? We would use the genotypic frequency equation, and all we have to do is plug in the numbers, right? So we know what big A is, we know what little a is, right? We plug in the numbers in our equation. So it would be 0.43 squared plus 2 times the multiplication of 0.43 times 0.57 plus 0.57 squared. Right? Now we would use our calculator, go through, do the math. Right? If I simplified it all down, I would get 0.19 plus 0.50 plus 0.33. I can check by my work by knowing that it equals 1. Right? Which tells me what my genotypic frequencies are for each of my genotypes. Right? So it's a lot of steps, all right? But if you follow it in a logical order, you should be able to get there. And there are ways to check your work along the way, making sure everything equals 1. Right? So we know our genotypic frequencies of our deer, right? Because we knew the Hardy-Weinberg equations, and because we knew how many deer were white. Right? That's all we had in the beginning. Right? So in a population of 200 deer, 65 of them are white. That's all the information I was given. I can figure out the allele frequency, and I can figure out those genotype frequencies. All right. I know this is one of those problems where you need to practice it to kind of understand it more. So again, there are those practice problems up on Moodle, and at the end of this slide, there's actually another practice set. Right, are there any questions? All right, well, you get a chance to practice it right away because that's our content check. All right, so this is our content check. If you do not finish it in the next 10 minutes, 12 minutes here, which you don't, that's quite fine, I'm going to accept late submissions, right? You can turn it in up until Monday if that's what you need to do, all right? I just ask that you show your work. So in our content check, we have a population of butterflies, right? 40% of the butterflies are white, right? White is the recessive allele. I want you to figure out the percentage of butterflies that are heterozygous and the frequency of the homozygous dominant individuals. This may take a while, so if you're not done by 1:20, right, that's fine. I'll accept these as late as Monday. You can have the weekend to kind of mull it over or go to tutors on Sunday night, whatever you need to do. So if you think about your example, it's 65 out of 200. What did that come up? No, either total population. If I were to tell you that 40% are 40, number 40 of butterflies, then you would need the total population